Okie doke. Well, you have the floor if you do have any questions, but yeah, you have to be Tuesday, Thursday, right? You're Tuesday, Thursday.
Yes.
Okay. I feel like I should have this committed to memory by now, but I just don't. Um, did you guys get the Valentine's Day chocolates I left you guys?
Yeah, they were by the door.
Yes, yes.
Yeah. Thank you.
Yeah, no problem. I just wanted to make sure because I can't physically come on Tuesdays or Wednesdays because I do have class that like runs on those times, uh, at the very least 1 to 4. So I couldn't come, but I was able to come on Monday, and I did bring candy for Monday. So I was like, I'll just leave extra, just make sure to set it out. I hope it helped de-stress while you guys were studying.
Yeah. You said you're doing the CNA course.
Yes.
I do have a quick question about that because my advisor is suggesting that I take it over the summer.
Mm-hmm.
Is that all just in lecture, or you have clinicals for that too?
You get clinicals. Right now, it's a little bit up in the air because of COVID. Right now, because of the surge that we had, uh, how the class is scheduled is that the first 3 weeks are completely, they're classroom based. You don't go out to a clinical site. Also, because you do need your like TB test, you need certain vaccinations or titers done. But because of those things, you have to wait the first 3 weeks, and usually on the fourth week you get to go out to a clinical site, and that's usually a nursing home or a resident care facility. So what happened was this year, because of the surge, they weren't allowing us to go. Some places were having surges specifically in their residential home, and some places were scared of having it happen to them. So we weren't allowed for the entirety of February, but we got reevaluated and they said for March it's possible. So we're in talking terms with the places, but typically yes. Are you going for the nursing program?
Yeah.
What I would recommend doing is if you finish all of your prerequisites, hold off, because more likely than not, you're not going to get accepted, or more likely than not, yeah, you're not going to get accepted the first time. So the first time you apply, it's kind of just getting your your feet in the water, testing the waters. So what I would recommend doing is taking the full 16-week course instead of just the I'm not sure how many it is for summer, but I know it's going to be faster over summer. And one of the big things is that I'm glad I'm taking a 16-week course because it gives me more time to memorize the items, and I don't feel like it's all being crammed into one, and I don't feel like I won't be ready when the time comes to take that standardized test. So if you're waiting, if you can get it over summer, great, do it. Then you can work as a CNA, you can get the experience, and I think the CNA offers you a path to see if nursing's for you. Some people do CNA and then they wind up never applying for the nursing program ever again because they said, this is not for me. And that's okay. Sometimes people are like, I want to be a tech. It just depends. But yeah, I think it's a great program. And if you're ready for it, do it.
Okay.
I know it's not so glamorous wiping people, but I feel like it's a little bit of a compassionate act, You can't really think of it as being gross. You have to think of it as like, if you were in that situation, it's embarrassing enough you can't wipe your own booty, so you can't make it like if you think about it differently, as in like this person's embarrassed it's even happening to them, don't make it worse. It kind of changes your outlook on it.
Yeah. I wasn't really super concerned with that aspect of it. It's
Okay.
more of being able to get a little bit of experience as to what it is like being somewhat in the nursing field.
Oh yeah, it gives you a good head start on vitals, um, bed changes. You'll have to chart. That's a big thing with a nursing assistant is charting and reporting. The hierarchy of the whole floor, working one-on-one with residents. It'll give you a lot of experience, and a lot of our professors or like teachers in the clinical side of things, they are nurses themselves and they work full-time as nurses, and they say that they understand and they can see when somebody has been a CNA before they go into the nursing program and when somebody has not. So I think that speaks volumes.
Yeah.
But yeah, I would recommend it. It makes you feel a little bit more prepared, definitely. Plus, it's a good backup plan. You know, you'll always have somewhere to work as a CNA.
That's very true.
So if you can't be an RN for so many years, but you still want to work in a hospital or you still want to work with patients, then being a CNA is great. But yeah. And also because of the recent grants that they've gotten, it's not as expensive anymore. You used to have to buy this book and all of your tote, which is like your blood pressure cuff and stethoscope. You used to have to buy all that on your own. Not anymore. That's at least a good couple $100 you don't have to spend anymore. The only things you really have to buy or like spend money on is the exam fee and the drug test you have to take at the beginning of the semester, and then any other medical things like if you go to the doctor, you have to pay a copay in order to get a physical. But the BC Health Center offers physicals and a TB test for free. So you can skate by with those things for free. But I gotta check. Yes, I do want to be a registered nurse. I would like that. And I got to talk with a registered nurse. She's in the 3rd semester pediatric rotation for nursing students. And she gave a lot more insight, and I'm actually like, it heightened my interest even more. So yeah.
That's awesome.
There is it takes a special kind of people, I think. And she said that she saw that too. And one of her things that she saw was when COVID hit, it was some of these nurses just dipped. Like it was not long into the pandemic. They said, I'm not doing this anymore. So there is a need for nurses. And because of that grant mid-COVID, I'm sure the impact was felt early on. And not only that, but Dr. B actually is working on a project to get a rotation in for the nursing program. That's a mobile unit. So this mobile unit is supposed to go out to more rural areas like Tehachapi, supposed to go out to certain places in, I think, Arvin too. You know, certain, like if you're thinking absolutely in the middle of nowhere, that's where this rotation is going to be. She's working that into the nursing program, and I just found that really interesting. I mean, you know, I think mobile units is it's going to be something that we definitely will need for the Bakersfield area. Even if it isn't the rural aspect of it, at least, you know, getting nurses to a certain area that maybe is understaffed, you know.
Yeah.
But yeah. Oh, thank you, Praise. But yeah, it's all interesting things, but enough about me. How do you guys feel about this exam?
Scared.
I promise that once you get into the thick of it, you learn how to chop down the weeds.
Did you, when you would do your exams, did you do the lab portion or the exam or the lecture portion or the, yeah.
What I found was that if I were to be, because I'm a visual learner, I have to have a little bit more hands-on. If I do the hands-on aspect first, so I always did lab first, I felt as if I had room to do things because not a lot of people do lab first. I felt like I had the room to do things, and I felt like it jogged my memory to see some of the graphs that she would provide for us to interpret. So when I went over to the lecture portion, I would be able to go in and better visualize those questions, if that makes sense.
Yeah.
But if you're more of the person that's like, I'm gonna get burned out, I would recommend doing it like whatever's going to take the highest energy first, because you don't want to do all these tedious multiple choice questions and then run out of energy, get to your short answer questions and go even slower, and then have to do the entirety of the lab section.
Right. I'm just worried doing the lab section is gonna, because when I went in and did the gram stain and streak for isolation, I was there for the whole 2 hours.
Mm-hmm.
So I don't want to, yeah, I don't want to end up running out of time.
But I'm sure during that time you probably asked questions, you referenced maybe something, you had to look through a book, or you know, you were taking your time, right?
Right. Getting all my stuff together.
Yeah. So when it comes down to the actual exam, all the stuff's going to be at your table, maybe except for tape, but all of the stuff is going to be right there. You just need to throw it all together.
Okay.
So it's not like, oh, I need to go over here to get something. Oh, I forgot it. I need to go get it. You'll have everything there, including a Sharpie, I believe. If not, tell her and she'll get you one. But it's all going to be right there. All you really need to focus on is doing your technique and doing it well.
Oh, can you go over the, you said when you streak for isolation, you do so many swipes in one, and then when you go to 2, you hit it 1 3 times or something like that, and then
Yes. So let me,
3.
this is my whiteboard that I use because I find it easier to show. Okay. So let me, come on, I just want to select something. Okay, I'm just going to draw it then. That is a, wait, I always forget this. I always do. I always forget that my computer screen's a touch screen. I always forget that, and then I try to draw with my mouse. I'm like, why is it so crummy? Okay, let me, that didn't happen, okay? I'm like Squidward erasing his picture in the Krusty Krab window. Okay. Yay. So this is my plate, right?
Yep.
When I do the bottom, when I label the bottom, that's going to be the true false. But when we, because it's going to be the lid of the dish and then the agar side up, when we open it, it's going to be backwards, right?
Yeah.
So when I'm looking into the plate, it's going to look like this, right? It's going to be the top section, quadrant 1. Let's write that in the corner. Our middle right here. And usually if you were looking at the back, it would go across to the right, but because it's reflected, we're going to go in here. So that's 1. This is 2. And if it helps you to draw it in the corner, great. 3, and then 4. Okay, so these are the quadrants, right? So for my first quadrant, I'm just going to paint it, right? I'm going to get it. I'm going to get my part of a colony, or if it's really, really small, like 1 or 2 colonies, I'm going to spread that nice and thick, right? Okay. So then now I'm going to go into my second quadrant. I want a different color, but I don't know if it will let me. Okay. Well, we'll make do. So when I go from the first quadrant into the second quadrant, I'm going to start in the first, right? I'm going to start in the first quadrant, and I'm going to go in 1, back up 2, 3, 4 times, right? And then I'm going to use the rest of this field to paint it out. I'm not going to ignore that I went back in, but I'm going to use the rest of the field to paint it out.
Okay.
Okay. And then once I paint it out, is this brown? No, it's not. I hope it's brown. No, nope, it's not. So once I paint it out, now I'm going to put the lid back down, and I'm in between each quadrant, I'm incinerating, right? So for the next quadrant, I'm going to go maybe 3 times. So I'll go 1, 2, 3, right? And then I'll use the rest of this field to paint it out. Okay. And then incinerate. And then for my 4th quadrant, I'm going to go 1, 2, right? I'm going to go in twice. I'm going to paint just a little bit, and then I'm going to do my Harry Potter squiggle.
Okay. One of the things that she said when I was, when I had my results, she said to maybe get a little less of the bacteria.
Yeah.
So I don't know if she means like just because it was really, really thick where I had got it, where I had gotten it from. So I don't know if she just barely touch it or.
So quick question, what bacteria did you sample, or was it the unknown?
It was the, I believe it was Bacillus cereus.
See, those always, so you saw the difference between B. cereus and maybe E. coli, right? B. cereus has some really big colonies. Yes. And I'm sorry, my chocolate chip. B. cereus has huge colonies, and if you were to try to take a whole one of those, you would never be able to isolate that. But if you were to take maybe like just tap the surface of it, you would have plenty of bacteria. Okay. But E. coli, you know, barely tapping the surface of one of those colonies could mean taking a quarter to half of it, right? Yeah. But if you look at P. aeruginosa or S. aureus, maybe that's not the same, right? It just depends on the colony size. But you don't want to grab too much. What I would say, it's hard to give like a right amount, but if you were to be looking like here, I'll draw it off to the side here. If you're looking at your loop, I hope you can see that. If you're looking at your loop and you're over a colony, try to grab a quarter of the loop. So like, maybe that much, even that might be too much.
Okay. So
like what I would suggest doing is aiming towards the edge of a colony. So if the colony is like this big and you have your loop, try taking that.
Okay.
But she said that she's not going to make you streak for isolation on B. cereus, at least on the exam. But later on, you maybe won't get that luxury for your unknown project, because that's going to be something that nobody knows, right? For me, I had the unfortunate event of getting a swarming cell, which meant that my entire plate was covered in bacteria and I was not able to isolate it. Because, you know, the swarming cells have flagella. They can move. It didn't matter what my technique looked like. It was, they just decided to move. So for when I had to do some things for like the gram stain, I had to just barely tap the surface. There was no colony to pick from. I just had to tap.
Oh gosh, okay.
But I don't know if you'll get a swarming cell. I was one of 2 people that had a swarming cell, and there was like 40 scenarios out of 20 people, so it was very niche. Let me see real quick my things, but that's grabbing less bacteria will do you good. If you were able to isolate in quadrant 2, that works. But you don't want it to be so thick that you can't even isolate in quadrant 4. Let me go to the chat really quickly. Also, get your rest tonight. If you have to wake up early in the morning, go to bed early. Wake up in the morning, treat yourself to a nice cup of joe, relax, study. Don't try to cram and stress yourself out, because it's only gonna, think about your energy, right? If you're cramming at 8 in the morning and you have to take this exam until 4 p.m., your energy is gonna dwindle. But get your rest, please. The question I have is, does 2 CO2 and 2 ethanol made from alcoholic, will that be made? Cram tonight and then review in the morning. But it just depends on how much you're putting in. So if the equation says that if you put in 1 glucose, you'll get so many of each of the end products, stick to that chemical equation. But I think all you really need to know for this exam is what goes in, what comes out, and why. So what goes in? Glucose, and through the metabolic processes, the Y of yeast, it ferments, which produces ethanol and CO2. So yes, so I hope that answers your question. But yeah, the streak for isolation should be okay for you guys. Just always remember, less is more, because yeah. Would it still be right if it was pyruvate instead? Not necessarily, because the whole process of fermentation includes glycolysis. So pyruvate is what is like the middle step, right? It goes into the fermentation cycle, but what really starts it is that glucose. Yeah, it's in the middle. So if she says, what are the beginning products of fermentation, you'll say a 6-carbon sugar or glucose, or if it's a specific to a fermentation cycle, say that. But yeah. Aw, I know. If you're having trouble with the lab portion, look at the lab study guide. And for lecture, I would recommend going back and redrawing diagrams, any absolutes like equations, write those out. Watch some crash course videos. There are actually some pretty good ones out there for those, for some fermentation stuff. Review videos are always nice. But yeah, I mean, usually I also sent out those practice quizzes. Praise, did you get those? I sent out 3. Oh, okay. I'll send them to you. Jamie, do you have them too?
I don't think I got them.
No? Shoot. I'll send them out right now. And then you guys let me know if you guys can see them. They're just some like lecture questions. It's not going to be exact questions on the exam, but they might cover topics that are on the exam. So let's see. I hope I'm not a terrible texter to you guys. I know sometimes I'm really critical of like some staff sometimes, and then I do the same thing because I think people would like me because of it, and then I'm like, no, I'm being cringe. Okay. Are you guys getting these? I think I've already sent out 2. And then this is the last one that I have. Okay. So, so those are the resources I have for you guys. If you want to go into the, like for an example that I have, I hope you guys can see when I pull this up. Can you guys see the PowerPoint?
No, I still see the auger plate.
Okay. There we go. Sometimes I don't share my screen. I just share a specific tab, and that's my downfall. So, for example, here, I could look at this and I have to ask myself how many molecules of ATP come out for each one of these steps, right? I can ask myself questions like that. Other theory portions might be, what does gas tell me from a F tube, right? What does it mean if it's pink? What does it mean if the MSA plate's yellow? So for your question, but for the question here, yellow would mean that there's an acid byproduct, so it did ferment the sugar. And gas would mean that it just produced gas in this pathway.
Okay, for some reason I was thinking it was the opposite. I knew that the yellow meant that it was acidic. I didn't know that so even if you have any gas bubble, it's still a fermenter?
Yes.
Okay.
Because it produced that acidic byproduct.
Okay.
I just think of the phenol red. If you ever hear phenol being in anything, it's a pH indicator. Think of it like a pH strip. It's going to change color if there's a change in pH.
Okay.
And then, quick question, what is the 4 components of an F tube?
The phenol red,
Mm-hmm.
the tube.
Mm-hmm.
Was it glucose?
It could be glucose or what other type of sugars?
Was it was that from our lab, sucrose?
Glucose, sucrose is one. And then one more. Milk.
It was lactose, yeah.
Yep, it'll contain a sugar type, right? So that's 3. You just have one more. Praise, do you know it?
What was
The what is the last component of the F tube? So right now we have the phenol red, we have the Durham tube, and then we have the sugar type, which is glucose, lactose, or sucrose. There's one more.
The pH indicator?
That was the phenol red.
Okay, let me see. Don't tell me, let me see. So I have no idea. I don't know.
Okay. I'll give you a word hint. Alternate.
No, not.
This also starts with an A. The two letters starts with A for both words.
Don't tell me. Is it amino acid?
Yep, amino acids. Those amino acids are the alternate food source for the bacteria. So that's our 4th and final component. I know, I always forgot that one too.
So proteins are okay. So proteins are okay. Part of the ingredients to basically identify if such fermentation occurs.
Yes. So, if we see a yellow, that means the bacteria really like that sugar and it fermented it and it created an acidic byproduct. sometimes it'll also produce a gas product.
Okay.
But also, if it didn't change colors, or if it's a negative for fermentation, that could mean it created a deeper red, became more basic by using the amino acids.
Okay.
Because bacteria will find a way. If they don't like the sugar source, they'll somehow use something else. If they're desperate, they might use the sugar source. I know sometimes what we see with E. coli in those F tubes is if it's not a sugar source they usually like, and it's really hard for them to digest that sugar, they'll still try. There will be a little bit of yellow pH indicator on the bottom, but the rest will be pink. It's because the E. coli was desperate to use that sugar source. Like, I'm pretty sure it's glucose, don't quote me. It's either glucose or sucrose. It is too big for them to digest. So they will try their darndest to try to digest it and live. But bacteria will find a way, life will always finds a way. All right. Glad I quizzed you on this. Yeah, what I would really suggest is, you see how there's this, it says metabolism, right? And then these subtopics. Write this down, and if you can't tell me everything about them, go back and review them. And then, so like for this one, I would say microbial metabolism, and then I would write down the definition maybe. And then obtaining energy, and then I would draw out this chart. I would write this down, and if I can't tell you what each of these mean or what each of these do, then I would have to go back, review it, maybe also write it out. Any definitions, that's a good one. Let's see. I would have a topic for enzymes, and then I would go here. I would write this out, understand how each of these influence enzymatic activity.
For that one, the pH. Okay, so like I know that a high temperature is gonna denature the proteins. I think the 35 degree, that's like the maximum point that the protein can ever, well, the enzyme can ever do its activity before being denatured. But for the pH, that's like a bit, because pH, if you have low pH, that's like a lot of hydrogen ions, a lot of protons, and that could pretty much mess up the nonpolar part of the proteins, which is like the inside. But my question was, if you have a low pH, I'm thinking that it's gonna denature. But if you have a high pH, like if it's basicity, like too much basic, would that also denature the proteins?
So let me see. Let me find that answer for you. So
If you don't know, it's fine, because I asked Professor Bake, and she said that like it's depend on the proteins. Like some proteins, it can be much stronger, so then therefore it can withstand, if it's low pH or high pH. I mean, which makes sense. But if it's a weak protein, then putting it in a high concentrations of hydrogen protons, it's gonna denature pretty fast. So I think she answered my question. I think it just depends on the proteins.
Okay.
Yeah, I don't think there's like a sure answer of that. Yeah, so
all for exam purposes, so that you're not stressing out over really in-depth ideas, because you did what I did. I was getting super granular, and I would go up to her with a question and she'd be like, no, stop thinking like that. You don't need to go that deep. Just that's it. And so sometimes you do have to take it at surface level and only critically think to a certain extent.
Okay,